messenger RNA. When these was allowed to undergo translation in the in vitro condition, they obtained several amino acids, and these amino acids were proline, asparagine, glycine, histidine, threonine, and lysine. Now, two codons, especially CCC and AAA, were deciphered in the beginning. So, rest of the codons might be coding for one of these four amino acids, but the exact meaning of this was difficult to derive out of such an experiment. So, the entire challenge of understanding the genetic code came to the end when ribosomal binding assay was developed by two scientists in the laboratories of Nirenberg himself. Now, this scientist, along with the help of Philip Leader, developed a process in 1961 called ribosomal binding assay. With these assay, the entire challenge of understanding the genetic code was conclusively ended. This experimental approach is based upon use of specific kind of filters called nitrocellulose filter. These filters are having a very fine porosity and has a tendency to retain large molecular weight substances, allowing small molecular weight materials to pass through. So, in other words, larger aggregates like ribosomes are retained over the membrane because they can't pass through the membrane, while small molecules like tRNA, amino acid, enzymes, and small proteins, messenger RNA, such small molecular materials can easily pass through the membrane. Now, what they have done is, as shown in this animation, he allowed messenger RNA and ribosomal aggregates to pass through the cytoplasmic membrane. And you can see that large aggregates like ribosome, messenger RNA complex were retained by the filter. Afterward, the messenger RNA attached to the ribosome were a small triplet of a known sequence, which were chemically synthesized in the laboratory, especially after the Hargobind Khurana joined the team of Nirenberg. The tRNA species which were charged with different kind of amino acid, when these was allowed to pass through the membrane, it will not be able to establish itself over the messenger RNA because the sequence specificity does not exist. But when tRNA, which is having a compatible anticodon arm, is allowed to pass through this membrane, it will establish itself to messenger RNA because sequence compatibility exists. And when this experiment is repeated 20 times, every time one of the amino acid was radioactively labeled, he was able to understand that if radioactivity is retained in one particular amino acid set, that amino acid is probably encoded by the sequence under the experiment. So, although it was a cumbersome and tedious task for every nucleotide tested, he has to carry out and run number of sets every time, starting with E. coli cells and preparing a fresh extract of them and a fresh cell-free system until the proteins are checked. But it eventually put an end to the challenging task of understanding the codons. And with these experiments and years of efforts by a team of scientists, entire dictionary of genetic codon became known to us. And you can see over in this chart that all amino acids are coded by one or more number of genetic codes. There are specific sequences as shown in the green called AUG, which codes for initiation of a process, and those sequences which are shown in red, UAA, UAG, and UGA, are responsible for putting an end to the translation process, are known as termination sequences. The rest of the codons are known as sense codons because they are coding for one or the other type of amino acid in particular. Now, how these genetic codes are read, what are the rules of this language, are there any punctuation marks or not, are these sequence overlapping with each other? A lot of questions remain unanswered. And the efforts were also going on side by side along with the understanding of the codon. And now I'll hand over to Piyush Bhai for explaining the general characteristic of this codon and the rules and grammars of the language spoken by nature. Piyush Bhai, please.
So once the clarity has been established, hopefully, among the students regarding the basic technology that brought to us knowledge the structure of the code and the constitution of genetic code, now let us capitalize upon certain characteristics which, let me make very clear in the beginning, that are universal. The characteristics of the genetic code are universal in the meaning that from a small bacterial cell to a plant or an insect or an animal to us, the human being, the genetic code remains almost the same. We'll be talking about that as well. But the characteristics by and large remain the same in all types of biological systems, be it a prokaryotic system like a bacterial cell, or be it a dinosaur, which though have become extinct, in their time would have followed the same rules and regulations regarding the genetic code's existence and its activities. If we may have the slide, please. The first thing that strikes us is that genetic code is made up of 3 letters, 3 nucleotides. What is the logic behind this? As we know, the nucleic acids, the messenger RNA which carries the message from DNA to the cytoplasm regarding the kind of protein to be synthesized, is made up of nucleotides, and by and large in a given nucleic acid we get 4 different varieties of the nucleotides. In a nucleotide structure, the sugar and the phosphate remain the same. The only variation possible is among the nitrogenous bases, and by and large, as we already know, 4 different types of nitrogenous bases, the nucleotides containing either adenine, which is called an adenosine nucleotide, and similarly guanosine or thymidine or cytidine kind of nucleotides are available. So we have at our disposal 4 nucleotides, out of which we have to prepare the codes. Codes will have to specify to the cytoplasmic protein making machinery what kind of amino acid has to be placed where. Now there are 20 different types of amino acids which are found in naturally occurring protein molecules. So 4 nucleotides have to give information for 20 different amino acids. Naturally, if we assume that the genetic code is made up of only a single nucleotide, then a single nucleotide code will be able to give information for only a particular amino acid, a single amino acid, and therefore only 4 amino acids can be specified. If we buy the fact that the code is made up of only a single letter, only 4 amino acids can be specified. If we assume that, okay, let the code be consisting of 2 nucleotides, then what will happen? 4 nucleotides, and every single code made up of 2 nucleotides at a given time, then the total combinations possible is 16. So still we fall short by 4 amino acids. 16 different genetic codes made up of 2 nucleotides can give information for 16 different amino acids, but what about the remaining 4? In that case, the next probability is that the code should be made up of 3 nucleotides, adjacent neighboring nucleotides. Given this, total number of codes available goes to 64. Now the problem is there are 64 different code combinations and we have 20 amino acids to specify. Now as we know, nature is a great economist. There is no extravagancy among nature. And if we go by this equation, for 20 amino acids, we have 64 different codes. So an extravaganza of 44 codes exists. And this is not acceptable. To the common sense of a scientist, this kind of extravagance is exorbitant and cannot be tolerated. But then the fact remains, as we'll go on talking about the characteristics of the genetic code, that this justifies the total number of 64 possible codes for 20 amino acids is quite justifiable. As a matter of fact, it is quite beneficial to a biological system. We'll be talking about that later. At the moment, let us have the next slide, which tells us the known overlapping property of the genetic code. As I told you only a little while ago, the genetic code is made up of 3 nucleotides, and these 3 nucleotides have to be adjacent to each other. The neighboring nucleotides on a given messenger RNA sequence, the 3 neighboring nucleotides make up a single code. Now, if we understand, let us assume for a little while that the code is overlapping. What do we mean by that? If you look at the figure, you can see a sequence of GCC, CAC, and if we by the fact that the code is made up of 3 letters, it gives us 2 genetic codes, GCC and CAC, where GCC, as you can see in the diagram, codes for alanine and CAC codes for histidine. Okay, if we accept that the genetic code is overlapping, then what will happen? In this sequence of 6 nucleotides, we can have a possibility of 4 genetic codes, the first code being GCC, then the next can be CCC, then the next can be CCA, and the fourth one can be CAC. Thus, as I told you a little while ago, nature is a great economist. It can conserve here by making a compact code structure where the 6 nucleotides in this figure can give 4 different codes. This is what an overlapping code is consisting of. But the fact remains that there is no overlapping. There has to be a demarcation, there has to be a differentiation of 3 nucleotides each, and there has to be no overlapping. This was proved very easily by an experiment where a change in any of the single nucleotide in a given sequence would affect only a single genetic code. If there was overlapping, for example, look at the third nucleotide in this given sequence, GCC. If this C is replaced by some other nucleotide, for example G, then the first code changes. GCC now turns into GCG code, and this gives information for tyrosine. So alanine will be replaced by tyrosine. Now the next code also should change. If the genetic code is overlapping, then the C being replaced by G will change the third code as GCA, and this will change another amino acid as well. So a single mutation, a single change in the nucleotide sequence will at a given time make 2 changes in the polypeptide. 2 amino acids would be replaced by undesirable amino acids. This doesn't happen, and therefore it has been confirmed, it has been established that code is never overlapping. There are certain exceptions in viruses where some kind of overlapping is done, but as we understand, they are rare exceptions. By general rule, in almost all biological systems, the code remains non-overlapping. The next slide tells us about the start and the stop signals. Messenger RNA is actually made up of a reading frame. It is called an ORF, the open reading frame, where the stop signal, first of all, the start signal is there, where the ribosomal assembly understands that this is the point from which the initiation of translation has to be made, and then there is a stop signal as well, where the whole process of translation, the synthesis of a polypeptide comes to an end. The initiation codon in most biological systems is AUG, and it always places as the first amino acid of a polypeptide methionine. The protein's translation machinery has been so established that in any given polypeptide chain, the first amino acid to be placed is methionine. In prokaryotes, this methionine is formulated. In eukaryotes, the formulation may or may not take place. We will not go into details of that. But in most biological systems, as a preliminary, as a primary genetic code, we have AUG, and that always invites methionine as the first amino acid of the polypeptide chain. Similarly, when the chain has to terminate, the whole process of translation has to come to an end. For this, there are three different genetic codes. These codes, UAA, which is called Osher, UAG, which is called Umber, and UGA, which is called Opal. It is also called Humber. They have a very specific function of bringing termination of the polypeptide chain synthesis, translation process, and thus there is no tRNA which is specific for them. They never invite the amino acids for translation machinery. Rather, they tell the translation machinery that this is the point from which the process has to terminate, and the ribosomal assembly has to be separated from the messenger RNA. So, let us remember that basically there are 64 different combinations of genetic codes, out of which 3 do not code for any amino acids. So, for amino acids, the SANS codon number is now reduced by 3, 61. We will be talking about how this 61 number is justified for 20 amino acids later on. But at the moment, it is very important to realize that out of 64, 3 have a very important function of bringing the polypeptide synthesis to termination. The next slide tells us about the degeneracy of the genetic code. Degeneracy or the redundancy is a feature of the genetic code where a single amino acid can be encoded by more than one genetic codes. And this tells us exactly why there are 61 codes for 20 amino acids. By a simple logic, the messenger RNA and the DNA molecule, which is responsible for synthesis of messenger RNA, are always in a dynamic state. That is, the DNA is always being threatened by a phenomenon called mutation, where the nucleotide sequence of DNA can undergo